So when you had to leave, it wasn't like something I said, was it?
Honestly, I was like talking and I knew the drink, and I was like, oh, that's me. On Friday, I'm missing the class because I have to go on the roof.
Okay. Yeah, we always lose a lot of people for that. It's a fun class, though. There's two things I want to do today. First of all, I want to finish looking at photosynthesis. We shouldn't take us too awful long. And then the second thing I want to do is start to review the major biochemical processes we've looked at, showing you sort of a flow chart of everything that we've talked about and the things that you really need to know for the upcoming exam, which is next week. Okay, so hopefully we'll be able to get through that. You'll remember last time I was talking about cyclic and non-cyclic electron flow as the two major systems that are used in the so-called light reactions to generate ATP and NADPH that can then subsequently be used to make glucose. The cyclic electron flow is used mostly in bacteria. It's just a situation where you pop electrons up to higher orbitals, you snatch them away, pass them through an electron transport chain, and then just return them back to the original photosystem. But in the process, you're pumping hydrogen ions across the thylakoid membrane so that you can drive ATP formation. In non-cyclic flow, which is shown in this figure, however, it's quite a bit more complex because not only are you making ATP, but you're making an ADPH. Cyclic electron flow only makes ATP, and you're using both photosystems. So the thing to do is just to go through this figure piece by piece in a stepwise fashion so we can see how everything is happening. So we start with photosystem 2. The first step is that two photons here are absorbed on the left by this antenna complex. The energy is popped back and forth through these pigment molecules. It's just being fluoresced, basically, until it finally hits this P680 reaction center. Okay? When that happens, the magnesium atom in that chlorophyll molecule has two electrons that pop up to a higher orbital, and those are captured. All right, so two electrons are sent up to a higher orbital. They are snatched away by the primary electron acceptor. In the meantime, what's happened is we've got an electron hole here. We're missing two electrons, and this reaction center is so energetic that it can take water and steal those two electrons away to produce oxygen gas and hydrogen ions. Right? So that's what happens. Two electrons from water come in, replace the electrons that have been lost, and that produces hydrogen ions and an oxygen atom. That oxygen atom is not, in a sense, comfortable being by itself. It'll quickly interact with another oxygen atom to form an oxygen molecule, which is then released by the plant cell. Okay, the next step then is to pass those electrons down the electron transport chain. This is no different than what you saw in the mitochondrion. It's almost exactly the same process. Many of the protein components are the same ones that are used in mitochondrion. Right? So ATP is produced by chemiosmosis. You pump hydrogen ions across the membrane, let them flow back. That allows ATP synthase to make ATP. Right? So that's how our ATP is produced. Those two electrons then are passed down to photosystem 1. And photosystem 1 then absorbs two photons. And just like in photosystem 2, those photons are passed from one pigment molecule in this reaction center to another until they hit the reaction center. Once that happens, then two electrons are popped up to higher energy. They're captured, passed down the electron transport chain, and picked up by NADP to form NADPH. Okay, so the two electrons that the P700 system loses are replaced by electrons coming from photosystem 2, and photosystem 2 replaces those electrons by snatching them away from water molecules. In this way, we've successfully made both ATP and NADPH that can be used in the dark reactions. This is a diagrammatic view of how the whole thing is working in the thylakoid membrane. And it's very similar to what's happening in the mitochondria. And here's the antenna molecules that are absorbing light. Those electrons are popped up to higher energy, and since they're very close to this electron acceptor, they get picked up, passed down electron transport chain, and that is used to pump hydrogen ions across the membrane. Light is absorbed by photosystem 1. Electrons are popped to higher energy, passed down electron transport chain to produce NADPH. That's where those two electrons eventually end up. In the meantime, in the thylakoid compartment, we've got water that's busted up to produce oxygen and donate 2 electrons back to this reaction center here. All right, so this whole thing is happening in the thylakoid membrane. In the meantime, we've got hydrogen ions, a gradient of them in the thylakoid compartment, and those hydrogen ions are then used to pass through ATP synthase so it can use the energy to make ATP. Right? Same thing, basically, that's happening in the mitochondria. Same conceptual framework there. Right? The ATP synthase pretty much works the same, too. All right, so that's what happens in the light reactions. You use light energy to pass electrons around to generate ATP and NADPH. The Calvin cycle is where that energy is actually used to produce components of sugars and a lot of other molecules as well. Okay, the ultimate output is a 3-carbon molecule, glyceraldehyde 3-phosphate. And if you've really studied the Krebs cycle and glycolysis, you'll remember that glyceraldehyde 3-phosphate is one of the breakdown products in glycolysis. All right, so to make a 3-carbon molecule, you won't have to remember glyceraldehyde 3-phosphate. Don't worry about that name. Just remember, it's a 3-carbon molecule, and that's used to make a whole wide variety of organic molecules, glucose and other sugars. The inputs are carbon dioxide. That's the carbon source. That's where the carbon is coming from to assemble these 3-carbon chains. Right? They just don't come out of thin air. You've got to have the carbon there originally, and that comes from carbon dioxide. The energy that's necessary for this reaction is from ATP and NADPH. Right? It is a cycle, so like the Krebs cycle, the starting materials are regenerated, so it just cycles around again and again and again. It's much more complicated than the Krebs cycle. In fact, a lot of cell physiology or plant physiology classes don't even bother with the steps in the Calvin cycle because they are very complicated. Right? We will just do a brief overview of the processes here. All right, so here's the overview. The inputs are simple: CO2, ATP, NADPH. You run those things through, and the output is a 3-carbon molecule, G3P, and ADP and NADP. All right, so the energy is used in this process. There's an enzyme that actually does this bonding of carbon dioxide into organic molecules. That process is called carbon fixation. And the enzyme is called Ribulose Bisphosphate Carboxylase, or Rubisco. Okay? Most people remember Rubisco. If you say Rubisco, everybody knows what you're talking about. Rubisco is a neat enzyme, but it doesn't work very well. It's probably the most common enzyme in the living world. As much as 50% of the total protein component of a plant may be molecules of this enzyme, and the reason is because it's not very efficient. It's not a very good enzyme, and it causes some problems for the plant, as we'll see. One of the problems with Rubisco is that it'll use oxygen just as easily as it uses carbon dioxide. So if oxygen levels build up to high amounts, then this process actually goes in reverse and generates CO2 rather than generating G3P. All right, so that's a problem for the plant that has to be solved. Okay, in a sort of overview way, here are the things that are happening in the Calvin cycle. We have this 5-carbon molecule here. We have 3 of those. This is ribulose bisphosphate. And we bring in 3 carbon dioxides and break these molecules down into 6 3-carbon molecules. Okay, so we have 15 carbons, we bring in 3 more, now we've got 18. And that's the first step. The next step is an energy usage step where ATP is used, NADPH is used to change this 3-carbon molecule into glyceraldehyde 3-phosphate. If we're just looking at the carbon skeletons, these look identical, but it's the side bonds that have changed. One of those will be pulled out. Okay? That sort of is representative of those 3 carbon dioxides that came in. We now have 5 3-carbon molecules, and we've got to get back up to 3 5-carbon molecules. So there's a whole bunch of complicated steps that are involved, and that's what these multiple arrows indicate. There's several steps involved that use ATP to get these molecules back into the original form. So all you're doing in the back end of this cycle is regenerating your ribulose bisphosphate, your 5-carbon molecule that you started out with. If I were you, I wouldn't worry too much about these numbers. Okay? The main thing to remember is the inputs, CO2, ATP, NADPH. The output of each turn of the Calvin cycle is 1 glyceraldehyde 3-phosphate. Okay? Carbon counting, we don't need to really worry about that much at this point. All right, so here is sort of a grand summary of what's going on in all of these photosynthesis processes. In the light reactions, light energy is being absorbed by photosystem 2 with electron transport chains. Light is being absorbed by photosystem 1 to produce NADPH and ATP. One of the other inputs of that is water, and one of the outputs is oxygen. The ATP and NADPH are used in the Calvin cycle, where carbon dioxide comes in to produce glyceraldehyde 3-phosphate, which is used to make sugars, which can be used for a whole wide variety of different processes. Cellular respiration, you can use it to make the cell walls of the plants by making cellulose, by storing it in starch. You can use these molecules to make other organic compounds. You can make lipids from them. You can make proteins from them. Right, so there's a whole series of reactions that can occur to take these simple molecules and make them into more complex things. That Calvin cycle, though, uses the energy that was captured in the light reactions, and so the NADP and ADP come back and are used to regenerate or capture light energy once again. So it's a cyclical process. All right, keep in mind where these things are happening. The light reactions are happening in the thylakoid membrane. The Calvin cycle is happening in the stroma of the the chloroplast. There's none of this that happens in the cytoplasm of the plant cell. All right, there are different ways that plants can do photosynthesis. And the reason that these different ways have arisen or evolved is because Rubisco doesn't work very well. Instead of tinkering with the enzyme, plants have simply made more and more and more of it to try and keep the CO2 levels up and the oxygen levels down. Okay, so at high oxygen, what this enzyme will do is actually break carbon dioxide out of organic molecules, okay, using oxygen to do it. So generate CO2, and that's very bad for the plant. Another problem for plants is that, remember that thin section of the leaf that I showed? There's openings in the epidermis, stomata, that allow gas exchange to occur. Unfortunately, they also allow evaporation to occur. So you get a lot of water loss from those leaves. On hot, dry days, what happens is those guard cells around the stomata close. But that prevents gas exchange, so oxygen levels start to go up. If the oxygen levels go up, then Rubisco starts using oxygen instead of carbon dioxide, and that's very bad. So a number of plants have come up with some different strategies for dealing with this oxygen problem. Okay, this is the typical thing that happens with what we call photorespiration. Okay? What some plants will do is they'll take carbon dioxide and they'll bind it into organic molecules to produce 4 carbon compounds. And then what they do is they shuttle that stuff down to a different tissue layer in the leaf. Right? A tissue layer that is not as strongly exposed to oxygen because it's not someplace where light reactions are occurring. Okay, remember, the oxygen is being built up because of the light reactions. So if you move the carbon dioxide down to a different tissue layer where the light reactions don't happen, the oxygen levels are lower. So the Calvin cycle can happen normally. We call these C4 plants because they move that carbon dioxide in 4 carbon compounds. Okay, so C4. Some other plants do this at different times. CAM plants. At the very start of this slideshow, I showed cactus, which are classic CAM plants. Pineapples are CAM plants as well. And what they do is they take CO2 in at night. When it's cooler, the stomata open up. They don't have as much water loss. So they suck in lots of carbon dioxide and bond it into organic molecules to form 4 carbon compounds. Those compounds happen to be acids. Okay? Then during the day, when it gets really hot and the stomata close, all that carbon dioxide is released. So you get this huge increase in the amount of carbon dioxide present in the tissues of the plant. Okay? And as a result, it's able to do the Calvin cycle quite nicely because the oxygen levels never get higher than the CO2 levels. You can actually detect this by taste. If you eat a prickly pear in the early morning, it'll be much more acidic than in the afternoon because those acids are being broken down as the carbon dioxide is being released. Okay. So those are 2 alternate strategies for dealing with the oxygen problem of photorespiration. Your book finishes up with the topic of global warming, which is sort of appropriate. One of the things that plants do for us is produce oxygen. They produce carbon dioxide as well because they have mitochondria just as we do. Okay? But there's a slight excess of oxygen that's produced, and that oxygen is critical for maintaining oxygen levels in our environment. One of the things that is thought to contribute to global warming is carbon dioxide. And increases in carbon dioxide can result from reductions in the amount of plant material in the biosphere. Right? You reduce the amount of plants, there's not as many plants to get rid of that carbon dioxide in the atmosphere, and so it tends to build up. Right? So the question is global warming real? Well, let's define it first. It's a steady rise in the mean global temperature of the Earth. Right, so that's what we mean by global warming. There are lots of data that point towards global warming. There's 30% more CO2 in the atmosphere, right, since the Industrial Revolution. Since we started using things like coal-fired plants, we've just dumped tons of CO2 into the atmosphere. Right, so there's 30% more. Carbon dioxide is a greenhouse gas. What that means is that heat energy can get through it to the surface of the Earth. But when that heat energy radiates back out, carbon dioxide causes it to bounce back towards the Earth. It can't get out. Right? So that's what would contribute to global warming. There's been an increase in melting of the polar ice caps, although I just recently saw a report in Science that said that perhaps this is a normal trend rather than something unusual. Because by looking at ice cores in Antarctica, for example, you can sort of track warming and cooling phases in the Earth. And it may be that this is a cyclical thing that we're starting to see again. There have been very warm recent temperatures worldwide. Some of the warmest temperatures on record have been recorded in the past several years. So that's sort of interesting. Changes in climate patterns. Increase in arid areas. Deserts are increasing at an alarming rate, not only because of potentially global warming, but also because of agriculture. Okay? So there's a real problem there. All right. I guess the basic question is this: are you willing to wait around another 20 years and see if it's really real or not? I mean, it's clear that we've got lots of greenhouse gases in the environment. It's clear that we've got lots of CO2 in the environment. Why not do something about it now, before it gets too late? Yes, there are economic issues. Yes, if you force companies to have CO2 sweepers in their smokestacks, that costs them money, and maybe that costs jobs. And that's unfortunate. Okay, so you have to try and weigh that economic benefit against the disadvantage of what if global warming is real and we get a 1% melt in the polar ice and New Orleans ends up underwater, because that's what would happen. Okay, so that's the question. Okay, any questions on photosynthesis? All right, what I want to do now is start reviewing some of these processes. I'm going to use overheads to do this. Krebs Cycle Electron Transport thing. We'll start up here in the corner with glycolysis. And if you're writing this in your notebook, probably you want to put the sheet the long way, because this is going to take up the whole sheet, at least. Okay? So we've got glycolysis. That's the first thing that we have in these biochemical pathways. Glycolysis, of course, is a process where we take glucose, okay, and we're going to send it through two different sub-processes. We have an energy input phase and an energy payout phase in glycolysis, so we might as well separate them out. What I'm also going to do is carbon count here. So I'm going to draw these molecules just as carbon chains. We've got 6 carbons in glucose. All right, so there's our 6 carbon chains. With glucose, we have an energy input. All right, so there's several reactions that involve an energy input where we're actually using ATP. So we take 2 ATPs and make 2 ADPs out of those. We also use a couple of phosphates. And we end up in this energy input phase with a molecule that has phosphates on each end. Okay, there's our carbons. All right, so 6 carbons and 2 phosphates on each end. That happens to be fructose 1,6-diphosphate, if you're curious. All right. The extra phosphates are coming from phosphate that's in the cytosol. You don't use ATPs for those. Okay. All right, so there's our molecule. We are now going to go through the energy payout for glycolysis. So here's our energy payout stage. That energy payout generates a number of products. It generates 2 NADHs per glucose. It generates 4 ATP per glucose, but of course we used 2, so our net is 2. All right, so we could put that in parentheses, net 2. And we end up having 2 3-carbon molecules. Okay, pyruvate or pyruvic acid. Okay, pyruvic acid. All right, there's everything you need to know about glycolysis right there, basically. Right?
Up on your 4-chain carbon, or your 6-chain carbon?
Which one, the glucose or the fructose? Yeah.
Is that a P04 at the end, or is it just?
No, right here. Here we've got our 6 carbons, 1, 2, 3, 4, 5, 6. We do our energy input where we use ATP. We also use a couple of phosphates from the cytosol. And we end up with this molecule. We've got a PO4 phosphate group, 1, 2, 3, 4, 5, 6 carbons, and another phosphate group. Okay? It's fructose 1,6-diphosphate. Then the energy payout gives us 2 NADHs, a net of 2 ATPs, but it's a real 4 ATP. And we get pyruvic acid as the outcomes. Okay, those 2 3-carbon molecules. All right, there's glycolysis. Now, we have to take glycolysis, or the products of glycolysis, and go in 2 different pathways. All right? We have to decide whether oxygen is available or if oxygen is not available. So we'll have 2 arrows here. One where we have oxygen, and another one, which I'll deal with later, where we have no oxygen, no O2. If we have O2, we can then go into the grooming step. Okay, we take those 3-carbon molecules that we made in glycolysis. Right, so there's our 3-carbon molecules. From those, we make a number of products. We get 2 carbon molecules that are attached to coenzyme A. Okay, so there's 2 of those. We also get out 2 CO2, so there's where the other 2 carbons went. We also get out 2 NADH molecules. All right, so there's our grooming step. Take 2 3-carbon molecules, make them into 2 2-carbon molecules attached to coenzyme A's. Pop off those carbons as carbon dioxide, and in the process we make NADH. Right, there's our grooming. The next step then is to take these acetyl-CoAs. In fact, we should go ahead and name these. These are acetyl coenzyme A. And those are going to come into the Krebs cycle. Right? In the Krebs cycle, we take those 2 carbon molecules that are attached to coenzyme A, we run them through this cycle, and we end up getting out of this 4 carbon dioxides. Okay, if we run this thing these things completely through. Where in the Krebs cycle you get those CO2s isn't relevant for us. We don't need to worry about it. Okay? Worry about the inputs and the outcomes, rather than where each individual step, what happens in each individual step. What else do we get out of this? Well, out of this reaction we get 6 NADHs. We get 2 FADH2s. All right? And we also get a couple of ATPs out of this. All right, so what have we done? We've taken those acetyl groups, completely broken out the carbons from those acetyl groups, and gotten rid of them as carbon dioxide. We've captured the energy as high energy electrons and a little bit of ATP. I can tell people need to study more. Actually, this should be coming back to you. You should be thinking, oh, okay, 6 NADH, 2, that looks like FADH2 up there. I mean, I know my handwriting's crappy, but there's only 2 things where you make 2 of them in this reaction. I've already got the ATP, therefore, by elimination, that must be FADH2, right? Yeah. Well, that's okay. The exam is not for a week. I'm not surprised that it's not entirely crystal clear yet. Okay, there's the Krebs cycle. Now, there's a whole bunch of NADH and FADH2 that we've sort of forgotten about, ignored. We're not going to ignore that anymore. We're going to start using that. The NADH, okay, is going to come into the next step. Right, so this NADH that was used in or produced in glycolysis, we're going to use that now. The NADH that was produced in the grooming step, and the NADH from the Krebs cycle, as well as the FADH from the Krebs cycle, is all going to be funneled into the next step, which is electron transport. Okay, so there's our electron transport. The way I try to think of electron transport is by actually diagramming it with a membrane, because that's where everything's basically happening. So what I'm going to do is draw the inner mitochondrial membrane. Okay, so this is the inner mitochondrial membrane. Okay, we take all of these NADHs and FADH2s and funnel them into this process. So we have, well, let's see, how many is that? That's 6, 8, 10 NADHs. Okay, so 10 NADHs, which come into this process, drop off their electrons and become NAD+. We have 2 FADH2s that come into this process, drop off their electrons and become FAD+. Okay. And we have electron transport that generates a hydrogen ion gradient. All right, so we have movement of hydrogen ions across the membrane. The last step in this electron transport is the production of water. So we take oxygen, hydrogen ions, together those produce water. Okay, in fact, probably what we ought to do is we ought to put an arrow like this that shows electrons moving. Okay? So you remember electron transport. Right? These electrons are physically being moved from one carrier to the next in the membrane. All right. The last thing we need to do is mess with this hydrogen ion gradient. It's sort of pointless to have that unless we use it. Well, that hydrogen ion gradient is then allowed to go back through the membrane, and ADP is then used to make ATP. And you get around 34 to 36 of those. Okay. Or 36 to 38 as your book says. Okay. So somewhere between 34 and 38 ATPs. Depending on other conditions. All right, so there's the oxygen pathways that occur. The pathways that occur when you don't have oxygen are actually fairly straightforward. If you don't have oxygen, what we're going to do is we're going to take these 3 carbon molecules and we are going to send them through one of 2 pathways, depending on what kind of critter you are, what kind of cell type you are. These are both fermentation processes, and in the first of these we take 1 carbon off of each of those molecules as carbon dioxide, and we generate a 2 carbon molecule that has that's an alcohol. Okay, so this is ethanol. In the other pathway, we have our 3 carbon molecule that basically just rearranges a little bit to produce lactic acid. All right? Both of these are fermentation processes. Okay? The fermentation process that produces CO2 and ethanol, or the fermentation process that produces lactic acid. In both of these processes, the reason the cell does it is to be able to take NADH generated in glycolysis and change it back to NAD+, dropping off those electrons. All right? And that NAD+ then comes back to glycolysis. All right, so there's what happens if oxygen is not available. Alcohol fermentation or lactic acid fermentation. Okay? Be able to draw this from memory and you'll be in good shape. No, seriously. Be able to draw this from memory, you'll be in good shape. I'm absolutely serious. You may be thinking this is a daunting task. If you were in the majors level biology, you'd have to know every single step and every single enzyme and the molecular structure of every single intermediate in all the reactions. All right? So consider yourselves lucky. Right, there it is. Okay, yeah, we have time to look at photosynthesis and recap that. I do have originals of this, so what I'll do is I'll just post these outside my office, and if you want to make a Xerox of it, you can. I would ask that you please not take the original for a long period of time so that other people can get them. I'm not sure how well these will Xerox. I did them in that sparkly gel ink. It's really pretty, but it may not Xerox too well. I love those pens. They're fun. So I'll post them outside my office so you can come by and be able to get a Xerox of it. Photosynthesis. All right, well, we should be able to do this fairly easily because you've seen photosynthesis recently. We have 2 things we need to talk about with the light reactions. We have cyclic electron flow, and non-cyclic. So I want to look at cyclic electron flow first. Fairly simple. We're not going to worry about numbers here, so we don't have to try and remember, X number of ATPs or X number of NADPHs. Don't worry about it. Just remember the inputs and the outcomes. With photosynthesis, who cares about numbers? I don't. All right, so we have light energy that's coming down and interacting with a photosystem. It happens to be photosystem 1, which is the same as a P700 photosystem. Okay, I'm not even going to worry about drawing the antenna complex. Who cares? All right. We've got electrons that are popped up to higher energy. Sorry about that. That's an E minus. Okay, those electrons are passed down electron transport chain until they're passed back to photosystem 1, where they can pop back up again. All right, that process produces ATP by chemiosmosis. All right, same thing that happens in the mitochondrion. Passage of hydrogen ions across the membrane, they flow back through ATP synthase, you get ATP. Very simple. All right, so that's cyclic electron flow. Sort of interesting, but not terribly interesting from our perspective, because there are bacteria that do this, but plants don't. All right, so what we need to do now is look at the more complicated process of non-cyclic electron flow. All right, and here it's a little bit trickier. Now here I am going to worry about the antenna complexes. So we've got our photosystem 2, which is the same thing as P680. And we have all of these pigment molecules in here. And in the middle we've got this reaction center, which I'll color in. So we know that's the reaction center. All right, light is absorbed. Specifically, 2 photons are absorbed. Those photons are passed from one carrier or one molecule to the next until they finally hit the reaction center. At that point, 2 electrons are popped up to a higher orbital and snatched away by an electron acceptor. They are passed down an electron transport chain, which generates a hydrogen ion gradient, an H+ gradient, to make ATP. Okay, chemiosmosis again. In the meantime, we've got an electron deficit here in photosystem 2, and so we take water. Well, we don't, plants do. Take water, bust it to get those electrons, and we get oxygen out of there. All right, so that's where the oxygen comes from. These electrons that have been passed down this chain have to be passed off somewhere. All right, and they get passed off to photosystem 1. So again, we need to draw our antenna complex with our pigment molecules and a reaction center right in the middle. Okay, so those electrons then are passed off to photosystem 1. In the meantime, light is absorbed by photosystem 1, and that light energy is passed from one molecule to the next until it finally hits the reaction center again, and 2 electrons are popped up to higher energy, just like in photosystem 2. Okay, those are passed down electron transport chain, where NADP is then picking up those electrons to make NADPH. Okay, so NADP becomes NADPH. We should label this photosystem. This is a P1 photosystem, same thing as P700. Okay. Well, what about the Calvin cycle? It's a complicated process, but for the Calvin cycle, there's not a whole lot you're going to have to remember. Here's what you have to remember. CO2 comes into the process, and in a reaction that is catalyzed by the Rubisco enzyme, all right, so you need to remember that Rubisco enzyme. In this process, we're going to use ATP. Okay, so we have ATP being used, and we have NADPH being used to generate a 3-carbon molecule, which can then be used to make various organic molecules, okay, including glucose. And then the whole rest of this cycle is a process of getting back where you started, and we don't have to worry too much about it. All right? I mean, there's really all you need to know about the Calvin cycle. You take ATP and NADPH from the light reactions, use that energy to fix carbon, to bond it into organic molecules. Then you pull out 3 of those carbons as glyceraldehyde 3-phosphate. You use that to make things like glucose. And then you just use additional energy on the other side of this reaction to regenerate your starting material so you can bring more CO2 in. It's really that simple on a basic level. Memorize this and you'll be in real good shape. Okay, and I will see you Friday. I'm going to start genetics.